Hello, my dear friends and my dear students. Assalamu alaikum and welcome back to Talk Lesson for You. Dear friends, as we discussed in the previous lecture, we discussed about the nucleolus and its composition, right? So, let me introduce this lecture again. That we are discussing about the structure of the human cell, and we want to discuss it and study it with a big detail, right? So, in the first lecture regarding it, I covered about the cell membrane, then we discussed about cytoplasm, then we discussed about the nucleus and nuclear membrane, right? And then we discussed about the nucleolus. Now in this lecture I'm going to discuss about the DNA. What is DNA and what is the composition of DNA and what are the functions of the DNA, right? So let me introduce my lecture. I'm going to discuss about DNA. So the full form of the DNA will be the deoxyribonucleic acid. Let me mention all these. Deoxy, right? What does it mean? I will tell you in a bit. Deoxyribonucleic acid, right? Look carefully. First of all, why they are called the nucleic acid? So let me mention all those important points. Deoxyribonucleic acid, right? So first thing we should know that why they are called the nucleic acid, because they have acidic nature, right? And they are found inside the nucleus. That's why they are called the nucleic acid. Why they are called the nucleic acid? Because these are the acidic substances which are present inside the nucleus, right? As we know, if this is our cell, right, and inside the cell there is a round part that is called the nucleus, and inside this nucleus we have this nucleic acid, right? And we know that nucleic acid is of two types. Nucleic acid, right? Nucleic acid is of two types. First one is DNA, second one is RNA, right? The DNA and RNA, what are the differences? I will clear here, right? So look, on both of them have acidic in nature, a little bit acidic in nature, and both of them are located inside the nucleus, right? We don't say that all the RNAs are present inside the nucleus, because some RNAs are also present in the cytoplasm, right? But DNA is always present inside the nucleus in our body, right? That's why we say that these are the acids which are present in the nucleus. That's why we call them nucleic acids, right? Now let's move forward and write the full forms of both of them, so to have a clear difference between their RNA and DNA. Then we will move towards the DNA in a full lecture, right? So let me write here the DNA, and here I'm writing the RNA. I'm not going to discuss all the differences between them, but to know a little bit about them. So look carefully. This D means deoxy, right? Deoxy and ribonucleic acid, right? Nucleic acid, done. And here we have the ribonucleic acid. R means ribo, and then we have nucleic acid. I hope you watch the lecture till the end to have a clear concept of the composition of DNA. I will discuss it in detail. So look here. Here we have ribo. So first of all, what is this ribo? This ribo is ribose, right? Ribose. And this ribose is a sugar, right? And this sugar is also known as This ribose is also known as pentose, right? Why it is called pentose? Because it has 5 carbons, and if we look at the structure of this, here we have oxygen, then this oxygen is binded to carbon, right? So there are 5 carbons, right, in the structure here. So like this, here we have carbon, here we have carbon, this is carbon, and here is carbon. So this is carbon number 1, carbon 2, carbon 3, carbon 4, and here we have another bond, and here we have another carbon. So this is 5 carbon sugar, right? So we say 5 carbon sugar, right? So 5 carbon sugar which is present in RNA, right? This is called ribose, right? And this 5 carbon sugar which is present in DNA, this is called deoxyribose. Why? Because we know that at carbon number 2 and at carbon number 3 we have a hydroxyl group here and a hydroxyl group here, right? So look carefully, this is a pentose sugar, and let me draw another pentose sugar here also. So this is the oxygen, right? And here we have again our 5 sugars, 5 carbons, right? Like this, carbon here, carbon here, here carbon, and here, and this is our carbon number 5. 1, 2, 3, 4, and 5. I hope you are watching the lecture, right? Then at carbon number 3 we have a hydroxyl group, and at carbon number 2 we have only hydrogen. We have only hydrogen. So here the oxygen has been gone, right? So look, what is the difference between this sugar and this sugar? So this is also 5 carbon sugar and this is also 5 carbon sugar. So what is the difference between both of them? Here this sugar is present in RNA right, and this sugar is present in DNA, right? So what is the difference? We have said here the deoxy. Oxy means oxygen, right, and D means less, right? So the sugar which has 1 oxygen less than the sugar of RNA, right? Here we have RNA, this is RNA. RNA has ribose sugar, right? Which sugar? Ribose sugar. And DNA has deoxyribose sugar, right? DNA has deoxyribose sugar. It means that in the pentose sugar of DNA there is the absence of 1 oxygen, right? And in the pentose sugar of RNA, the oxygen on both carbon number 2 and carbon number 3 both are present. So this is the difference between the sugar of RNA and sugar of DNA. So that's why we call DNA as deoxyribose. Deoxyribonucleic acid. If we put 1 oxygen back here, so it will become ribonucleic acid, that will be your RNA. But if you take back that oxygen, that will become deoxyribose. So we will put this sugar in DNA, right? So in DNA, what is the difference between the sugar of DNA, sugar of RNA and DNA? So there is only the difference of 1 oxygen. The sugar of DNA is 1 oxygen less than the sugar of the RNA, right? So we have 2 types of nucleic acid. One is DNA and one is RNA, right? And what is the difference between them? There are many differences, but one difference in the sugars of both I have explained, right? So now you know that why we call it DNA. Na means nucleic acid, here also nucleic acid, but here DNA, deoxy, and you know it very well that in the in the sugar of DNA there is 1 oxygen less. That's why we call it deoxy. Deoxy. D means less. Oxy means oxygen. Right? Okay. So this was the full form of this. Now we move and define what is DNA. Right. Look carefully, friends, how our bodies are formed. Right? How is my body formed? Right? When my parents had decided to take a child, right, to produce a child, and that child was me, how was I produced? Right? From my father, right? From my father, his DNA came, and from my mother, her DNA came, and both of these DNA combined, right? both of these DNAs combined, right? And this DNA made me, right? Made me. Now how this DNA made me? Right? Look carefully. What happens when how do we form? How do living organisms form? Like this. Look carefully. The DNA makes RNA, right? The DNA makes RNA, and the RNA makes up proteins. RNA makes proteins, we know this. And there are 3 types of proteins in our bodies we have, right? These proteins will be structural proteins. Structural proteins. These proteins may be functional proteins. Functional proteins. And these proteins may also be regulatory proteins. Regulatory proteins, right? So look, from this DNA, the DNA which was given from my father and from my mother to the zygote, that DNA had came, and that DNA started replicating itself, right? DNA replication occurred. How? It made RNA, right? And that RNA was transformed into a protein, and those proteins, those RNAs were making 3 types of proteins. The structural proteins, these were responsible to make structures of my body, the tissues of my body, the cells of my body. And these proteins, the functional proteins, were certain enzymes, and these enzymes were helping these structural proteins to make these structures, right? So I want to say that how an organism is formed. Every organism is formed from its DNA. How that organism forms, this is the process by which our bodies are formed, right? How? Once again, look carefully. From DNA, right, every parent gives its copy of DNA to his child, to the zygote, right? When the DNA goes to the zygote start developing the baby, right? How? This DNA makes RNA, right? From certain point, for example, this is a DNA, right? And at a DNA, for example, for my skin, there are groups of genes, these black genes, right? And these black genes are going to make my skin. So how will they make? This part of this DNA will be transformed into messenger RNA. And this messenger RNA will be transformed into proteins, right? And those proteins will be of 3 types: the structural proteins, the functional proteins, and the regulatory proteins. And these proteins will start making my skin, right? So this is the way how the organisms are formed, how the body of organism develops, right? And now you know that the master planner of life is DNA. I'm sorry, I have a little problem. The master planner of life is DNA. DNA makes every organism. My DNA made me, right? The DNA which will go to my child, that DNA will make my children, right? The DNA of a carrot will make the carrot. The DNA of a rabbit will make a rabbit. The DNA of a tiger will make a tiger, right? So every organism is made from DNA, right? So we say that DNA is the master planner of life of all living organisms, right? So let me write its definition a little bit. The master planner of life, master planner of life, it means that life begins from DNA, right? Life begins from DNA. Now, what exactly is DNA? How can we define DNA? So DNA is made up of small subunits, and those small subunits are called nucleotides, right? So DNA is a polymer of nucleotides. The next definition of DNA, let me write here, polymer of nucleotides. Look carefully. Poly means many, mer means molecules, and nucleotides, it means that if we combine many nucleotides, if we combine many nucleotides like this, look, if we join these, right, so they will make a long molecule, right? They will make a polymer, and this polymer is called DNA. And what is this? This is a nucleotide. Nucleotide, right? So what do? We put together many nucleotides, and we make a long chain of nucleotides. That's why we call that long chain of nucleotides as DNA. So what is DNA? Long chain, long unbranched chain of nucleotides. The next definition, long unbranched, right, chain of nucleotides, right? So what do? We combine many nucleotides and make a long chain, and that long chain is called DNA. And as I have written here, the master planner of life. The master planner of life means every action which occurs inside our body is planned by DNA. Everything which occurs inside the cell is planned by DNA. Nothing happens without the permission of DNA inside our body, and if that thing happened, that will be a wrong thing, right? So DNA plans every action which occurs inside the cell, which occurs inside the tissue, which occurs inside an organ, right? So DNA is a very important thing for a cell. Okay, now let's do the composition of DNA. So the DNA is made from what? As I told you here, that we will make these nucleotides, and we will combine these nucleotides, and we will make the DNA, right? So look carefully, what the DNA is made from. DNA is made from nucleotides. Nucleotide. So what is nucleotide? Nucleotides are the building blocks of DNA, or we can say nucleotides are the building blocks of nucleic acid, right? So let me write here building blocks of nucleic acid. And this nucleic acid, you know that we have two types of nucleic acid in our body. That is, the nucleic acid may be DNA or it may be RNA, right? So it means that these nucleotides will combine and they will either make DNA or RNA, right? So there will be some differences between DNA and RNA we will see in the end, right? So first of all, let me make a nucleotide. Now in order to make DNA, we should have lots of nucleotides. Right? So let me give an example to you. If you want to build a wall, right? So you should have blocks. And none of the walls can be made without blocks. So in order to make a DNA, you should have nucleotides, right? So first of all, we should make nucleotide, then we will combine those nucleotides and we will make a long polymer, and that will be the DNA, right? So let us do that. So in order to have a nucleotide, to make a nucleotide, we should have three things. We should have three things. If someone asks, the DNA is made of what? DNA is made of three important things. First, look carefully. DNA is made up of three things. So let me name them. Those. First of all, we have a nitrogenous base. Nitrogen base. Right? Number two, we should have sugar. Right? And number three, we should have phosphate. Right? So these three things will combine and they will make DNA. Right? So look, first of all, let us know a little bit about these nitrogenous bases. Nitrogen bases. Nitrogen bases are also known as nitrogenous bases, and these are called nitrogen bases because in the ring of these organic compounds there are lots of nitrogen. That's why we call them nitrogen bases. So nitrogen bases are of two types, right? Nitrogen bases are of two types. One group of nitrogenous bases, we have double ring nitrogenous bases, and one group we have single ring, right? We call them double ring, right? Double ringed, and we call these as single ringed, right? These double ring nitrogenous bases are called the purines. Right? And these single ring nitrogenous bases are called the pyrimidines. Right? These are called the pyrimidines. Right? Pyrimidines. These double ring nitrogenous bases are called the purines, and there are two double ring nitrogenous bases, and these are called the adenine and guanine, right? So the double ring nitrogenous bases are the adenine and guanine, right? And the single ring nitrogenous bases are three, right? There is the thymine, then there is cytosine, and then there is uracil, right? So these three are the single ringed, and these two are the double ringed, right? Now in case of DNA, we have the adenine present with us, we have the guanine, we have the thymine, and we have the cytosine, right? This uracil will be present in RNA, right? So that's why we don't use uracil here in case of DNA. Right? Once again, in order to make the DNA, we should have many nucleotides. And in order to make the nucleotide, we should have these three important factors, these three important things. The first, nitrogen base, the second, sugar, the third, phosphate, right? What are nitrogenous bases? These are the nitrogen containing organic substances, right? There are two groups of them. The first one, double ringed nitrogenous bases, are called purines, and the single ring nitrogenous bases are called the pyrimidines. The double ring nitrogenous bases are adenine and guanine, and the single ring nitrogenous bases are thymine, cytosine and uracil. So thymine, cytosine, adenine, guanine, these four nitrogenous bases are present in DNA. But in case of RNA, right, we have uracil also. So uracil will be present instead of thymine. We will see that later while we discuss RNA, right? Then the next thing that we should have is this sugar, and which sugar should be there. And you know, in the beginning of this lecture, I told you that the sugar which is present in DNA is called the deoxyribose. Deoxyribose sugar, right? So which sugar? Deoxyribose. And we know the structure of that deoxyribose sugar. We know, and I have drawn it for you guys, right? And here we have our oxygen, and then here we had our hydrogen, and then here we had our hydroxyl group, right? So this was the sugar, right? And you know that the third and important thing that we should have is our phosphate, right? So we should have phosphate group, I mean PO4 group, PO4. So we will have this one also. Now what do? Now we combine these three, right? The nitrogen base, the sugar and the phosphate, and we will make one nucleotide. And in that manner we will make millions of those nucleotides, and then we will combine them and we will make the DNA, right? So see the next part in the next lecture.